Welcome to the deep dive. Today we're doing a high-yield run through the absolute core of biochemistry, molecular biology, and genetics.
We've pulled all the critical concepts straight from the $6 edition of BRS Biochemistry.
So this is really designed for anyone doing a rapid professional review. If you're studying for an exam or, just need a solid refresher on how all this stuff drives health and disease, this is for you.
Exactly. Our whole goal today is clarity and depth. We're going to build up from the structure of a single molecule, bit by bit, all the way to complex cellular pathology.
And make those direct connections.
Yes. We want you to see how concepts like, say, enzyme kinetics or protein structure.
Yeah.
That's the language you use to actually understand, diagnose, and ultimately treat clinical disease.
Okay, sounds good. So let's unpack this and start where all the action happens. The molecular machines, proteins and enzymes.
And we have to begin with the building blocks.
Amino acids. And specifically, their charge dynamics, which all comes down to one key concept, pico.
Pico is absolutely fundamental. It dictates the charge state, and that charge state dictates the function of any amino acid inside a protein. The rule is pretty simple.
If the pH is below a group's pico, the group is protonated.
And if the pH is above the pico, it's deprotonated. That's it.
So for most of your non-ionizable amino acids, the ones without charged side chains, you only have 2 pico values to worry about.
Right. You have the alpha carboxyl group, which is acidic, so its pico is down around 2.
And the alpha amino group, which is basic, with a pico up around 9.
But then you get the interesting ones. When you have an amino acid with an ionizable side chain, think histidine, lysine, aspartate, you suddenly have a third pico.
And that third one is essential for function.
That's the whole secret.
Yeah.
That third variable pico is exactly why those amino acids are the workhorses for acid-base catalysis in enzyme active sites. It's not just theory to memorize.
No, not at all. It's why drugs are formulated the way they are. The ionization state determines if they get absorbed better in the acidic stomach or, the more neutral intestine.
Precisely. It directly informs clinical pharmacology. This stuff is never purely academic.
Okay, so let's follow that chain from structure to function. The primary sequence, the string of amino acids, is basically the instruction manual.
It dictates the final, folded, three-dimensional machine. And if that structure gets corrupted, the machine breaks.
And there's no better example of that structural fragility than the prion protein.
The prion is such a profound and, frankly, scary illustration of structural failure. The normal, healthy form, which we call PRPC2, is a monomer.
And it's mostly alpha helix structure,
Yeah.
right? Something like 40%.
Exactly. It's stable, it's soluble, everything is fine.
So wait, the pathology isn't from a new mistake in making the protein, but from a complete overthrow of an existing one. How does that normal protein get converted?
It's an induced conformational change. The disease form, PRPC2, refolds into this extensive beta sheet structure. It jumps up to 40, maybe 50% beta sheet content.
And that new shape is the problem.
A huge problem. This refolded version is resistant to proteases, and critically, it promotes aggregation. These aggregates form plaques.
Which leads to things like mad cow disease, an infectious protein structure that just flips the whole script on disease transmission.
It really does.
Okay, that's a stunning example. So moving from structure to action, let's talk enzyme kinetics. Enzymes are catalysts, and their main job is to speed up reactions.
They do that by lowering the Gibbs free energy of activation, the delta G diagram.
And to measure that catalytic action, we use 2 key metrics, Vmax and $0.5.
Right, Vmax is just the maximal reaction velocity. It's what you get when the enzyme is completely saturated with substrate.
And the Michaelis constant, polymolars, is the substrate concentration you need to get the reaction to exactly half of that Vmax.
You can almost think of polymolars as a proxy for the enzyme's affinity for its substrate.
Okay, so if the diol is low,
It means the enzyme has a high affinity. It's really good at grabbing and holding onto its substrate, even when there isn't much of it around. A high Gauler means the opposite.
and because the plots of velocity versus substrate are these hyperbolic curves that are hard to read, we use the Lineweaver-Burke plot.
Indispensable. It just transforms that curve into a straight line, which makes finding Vmax and dollar graphically so much easier.
Which brings us directly to drug design, because so many therapeutics are just exploiting enzyme inhibition. Let's start with competitive inhibition.
Okay, so in competitive inhibition, the inhibitor molecule is competing directly with the normal substrate.
For the same active site.
Exactly. And the fascinating part is you can actually overcome this. You can beat the inhibitor just by flooding the system with more substrate.
And you can see that clearly on the Lineweaver-Burke plot. The lines for the inhibited and uninhibited reactions cross right on the
Right.
y-axis.
The Vmax stays the same because you can just overwhelm the inhibitor, but the apparent kata goes up. It looks like the enzyme has a lower affinity.
A classic example being malonate inhibiting succinate dehydrogenase.
Now, in sharp contrast to that, you have irreversible inhibition.
This is where the inhibitor binds and just permanently knocks the enzyme out of commission.
And this strategy allows for some incredibly potent drug action. I mean, think about aspirin.
It's an NSAID that works by irreversibly inhibiting cyclooxygenase, or COX.
Which then interferes with making prostaglandins and thromboxanes, those secondary mediators of pain and inflammation. Once that enzyme is blocked, it's done for. The cell has to make a whole new one.
The precision is just incredible. We see this in chemotherapy, too.
Absolutely. 5-fluorouracil, or 5-FU. It's a chemotherapy agent that works by shutting down thymidylate synthetase.
So you stop the synthesis of DTMP.
And you starve rapidly dividing cancer cells of a key building block they need for DNA.
Another great example is allopurinol for gout.
A classic suicide inhibitor. The enzyme, xanthine oxidase, actually tries to process the allopurinol. It oxidizes it into oxypurinol.
But then
the oxypurinol binds so tightly to the active site that it never leaves. It effectively kills its own assassin, permanently blocking the enzyme and cutting down uric acid production.
And just one last darker example, organophosphates. Things like malathion.
Right. These are irreversible inhibitors of acetylcholinesterase, the enzyme that breaks down the neurotransmitter acetylcholine.
So you get this massive unchecked buildup of acetylcholine.
A cholinergic crisis.
Yeah.
And the symptoms are severe. Bradycardia, sweating, vomiting, blurred vision, all of it stemming from one single molecular block.
Terrifying. Okay, we've seen the power of chemicals on proteins. Now let's pivot to the blueprint itself. DNA replication and the errors that cause genetic disease.
To replicate that blueprint, you need a whole molecular team. It starts with helicases, which unwind the double helix.
Then you need single-strand binding proteins to keep those strands from just snapping back together.
But the real stress managers are the topoisomerases. As you unwind the DNA, you build up this incredible torsional strain ahead of the replication fork.
And the topoisomerases relieve that strain by snipping the DNA, letting it unwind, and then rejoining it.
And that function is a fantastic clinical target. DNA gyrase, which is a specific topoisomerase in prokaryotes, is the target for quinolone antibiotics.
Ciprofloxacin.
Exactly. You inhibit the bacterial gyrase, you stop bacterial proliferation dead in its tracks. A perfect example of targeting a molecular difference between us and them.
Now, for the DNA polymerases themselves, directionality is everything. They read the template strand from 3 to 5'.
But they can only build the new strand from 5 to 3'.
Oh, and they can't start from scratch. They always need a little RNA primer, which is made by an enzyme called primus, just to get going.
Crucial point.
So once that DNA is copied and transcribed to mRNA, the genetic code is read. The codon, 3 bases, specifies an amino acid. The AUG start codon sets the whole reading frame.
Which raises the big question. What happens when that blueprint changes?
When you get mutations.
We classify them based on what they do to the protein. Let's start with a missense mutation. That's a simple one-for-one amino acid swap.
And the textbook example here is sickle cell anemia.
It's a single-point mutation. A GAG becomes a GTG in the beta-globin gene.
And that swaps out glutamate, which is hydrophilic, for valine, which is hydrophobic, at position 6.
And that one tiny change has a massive structural consequence. That hydrophobic allele makes the hemoglobin molecules polymerize and stick together when oxygen is low.
Which distorts the red blood cells, causing hemolysis and all the vaso-occlusive problems.
Then you have a nonsense mutation.
That's where a normal codon gets changed into a stop codon.
Right, so protein synthesis just stops prematurely. You get a truncated, usually useless protein.
And maybe the most devastating is a frame shift mutation.
Yeah, that's caused by an insertion or a deletion of bases that is not a multiple of 3.
So the whole reading frame gets shifted downstream from the mutation.
And the result is just a completely scrambled, incorrect sequence of amino acids from that point on. A total mess, like we see in hemoglobin wane.
These defects don't just hit globular proteins, they also hit the core structural proteins, leading to connective tissue disorders.
Let's start with a nutritional one that acts like a structural defect, scurvy. It's not genetic, it's just a vitamin C deficiency.
And vitamin C is essential for modifying collagen correctly.
Without it, your collagen synthesis is impaired. You get fragile blood vessels, bleeding gums, those little red spots.
Now, contrast that with a true genetic disease like Alport syndrome. That's a mutation in type IV collagen.
And that specific type of collagen is crucial for basement membranes in the kidney, the inner ear, and the eye.
So a single defect in one scaffolding protein cascades into kidney failure, hearing loss, and misshapen lenses. It has completely different organ systems.
Exactly.
And we see similar failures in the cardiovascular system. Familial hypertrophic cardiomyopathy, FHC,
That can be caused by a mutation in the beta-myosin heavy chain.
the contractile protein.
Yes. So that mutation messes up the sarcomere, leading to that characteristic thickening of the left ventricle. It's a heart muscle failure driven by one faulty protein.
So we've had structural protein failure. What about a systemic failure from a deficient enzyme inhibitor?
That brings us to alpha-1 antitrypsin deficiency. This is a protein that circulates in your blood, and its job is to inhibit elastase.
And elastase is a powerful protease that our neutrophils release to destroy bacteria we inhale.
So if you're deficient in alpha-hevanellin antitrypsin, that elastase just runs wild. It starts chewing up the delicate elastic fibers in your lungs, which ultimately causes emphysema.
And here's the direct link to lifestyle.
It's a perfect molecular link. Cigarette smoke contains oxidants that specifically destroy a key methionine residue in alpha-hevanellin antitrypsin. So smoking directly inactivates the shield you have, which dramatically accelerates the emphysema.
That is such a powerful connection, which brings us perfectly to our last section. Cellular metabolism and organelle pathology, starting with the powerhouse, the mitochondria.
Mitochondria are central. It's where we oxidize fuel and generate ATP through the TCA cycle and oxidative phosphorylation, or OXPHOS.
And we have to remember the efficiency difference between the two main electron carriers.
Right. NADH oxidation gives you about 2.5 moles of ATP.
But text FADH22 oxidation only yields about 1.5 moles. So why the drop?
Think of the electron transport chain, like a series of water wheels or pumps. NADH drops its electrons off right at the beginning, at complex Y. It gets to turn all the wheels.
Maximizing the proton gradient.
Right, but text FADH2 enters later on. It bypasses that first pump at complex I. So it just misses out on that first step of energy generation, which reduces the total yield.
And when the mitochondrial DNA, the mtDNA, is mutated, these energy problems become systemic, hitting the tissues that need the most energy: muscle, nerve, kidney.
A classic example is Kearns-Sayre syndrome. It's characterized by ophthalmoplegia, paralysis of eye muscles, and severe cardiac conduction defects, all due to failures in the cell's energy supply.
Okay, next organelle, the lysosome, the cell's recycling center.
And it has to maintain a very acidic internal pH around 5.5 using a proton pump.
If one of the lysosomal enzymes is defective, the material it's supposed to break down just accumulates. That's a lysosomal storage disease, an LSD.
Like Tay-Sachs or Gaucher disease. But sometimes the enzyme itself is fine. It's the delivery system that's broken.
The cell's molecular post office, that's eye cell disease.
Exactly. The problem is with enzyme targeting. Lysosomal enzymes are supposed to get tagged with a mannose-6-phosphate, or M6P, signal in the Golgi.
Like a zip code.
A perfect analogy. In eye cell disease, they don't get the M6P tag. So without the correct molecular address, the cell doesn't know where to send them, and they just get secreted outside the cell instead.
Let's finish by connecting all this metabolism to some common toxicities, starting with ethanol. The acute effects of alcohol come from that huge spike in the text NADD plus ratio.
As your body metabolizes the ethanol, it burns through tons of text NAD plus dollar. This high NADH ratio actually shuts down other key reactions, like fatty acid oxidation.
And the chronic damage, the hepatitis and cirrhosis?
That's largely due to the buildup of the toxic intermediate acetaldehyde and all the free radical damage it causes.
Okay, another critical one, acetaminophen overdose.
High doses just completely overwhelm the body's normal glutathione detoxification pathways.
And that leads to the buildup of a really toxic metabolite.
Text NPKO, the text, N-acetylbenzoquinidine, and without treatment, it causes massive liver necrosis.
The emergency treatment is text N-acetylcysteine. Why does that work?
Because text N-acetylcysteine is a precursor for making more glutathione. You're basically giving the liver the raw materials it needs to build up its defenses, conjugate the text NEPQI, and safely excrete it.
Okay, last one. Niacin deficiency, which causes pellagra.
Niacin is the precursor for the vital cofactors text NAM plus and destin ADP plus Nalazer. You need these for hundreds of reactions all across metabolism.
And a deficiency in these fundamental molecules leads to that classic clinical tetrad.
The 4 Ds: diarrhea, dermatitis, dementia, and eventually death. It's just a stark reminder of how one missing vitamin can cause total systemic failure.
This has been an incredible synthesis. We've gone from the charge of an amino acid all the way to complex clinical syndromes.
The key takeaway, I think, is to always think critically about the specific mechanism of action. The enzyme, the mutation, the cofactor. That's what drives the pathology, and that's the most crucial skill you can develop.
That approach leads us right to our final provocative thought for your review. This one connects diabetes, vitamin deficiency, and a metabolic crisis.
So think about the common denominators.
Given what you know about enzyme inhibition and type 2 diabetes treatment, a patient takes glipizide, which increases insulin secretion. If this patient accidentally double doses, causing severe hypoglycemia, why does this crisis require not only glucose but also careful monitoring of alpha-ketoglutarate dehydrogenase activity if they also suffer from beriberiya thiamine deficiency?
So to figure this out, first remember what glipizide does. It forces insulin secretion no matter what. Second, think about what the cells must do for energy when there's no glucose. And finally, consider which critical enzyme complexes rely directly on thiamine pyrophosphate. Good luck connecting those molecular dots.